Next thing is multi-photon microscopy. So where light sheet was very much a, we're looking at tissue, it's fixed. In multi-photon microscopy, we can look in live and viable model organisms. So you'll recognize the Jablonski diagram on the right-hand side here. And thus far, I've told you that the photon of light is collected by the fluorophore, and that does the transition. In multi-photon microscopy, what we're using is wavelengths of light, which are much, much longer. So about double what you would use for what we call a single photon experiment. And these photons are pulsed at a very high rate. And what we're trying to do is actually get two photons to be absorbed by the molecule at the same time. Although they're a longer wavelength with less energy, if you get two being absorbed at the same time, they basically add up to get you to that excited state. Now, what's the point in doing that? So the nice thing is that the longer wavelengths of light can penetrate deeper into tissue, and that allows us to do imaging live within tissue. So you'll see here examples of single photon excitation, which is what we talked about so far, and then the two photon excitations that are used for the same molecules. This is an example again taken from a lab here at the University of Melbourne, where they're looking at two different types of killer T cells in response to an infection with the herpes virus. So in red, they've got non-herpes specific killer T cells, and in green, they've got the herpes specific killer T cells. And what they're doing within this experiment is trying to track differences in the mobility of the different cell types. And again, this is all done in the skin of a live anesthetized mouse. It's a very, very useful tool in terms of understanding immunological cells live within samples. We do regular experiments on skin, brain, lymph. We're now switching into retina as well if people are interested in those applications. The next thing, so back to dead samples. Fixed samples is a slide scanner system, which is fairly new into our platform. So this is very much an automated and high throughput imaging of fixed samples. So either tissue sections or cells, but essentially they have to be on a slide and cover slip setup. We can image in either the bright field or multi-channel fluorescence. Our system that we have in the platform is the Zeiss AxioScan 7, which can do 100 slides at a time. So we have 25 cassettes here, which load 4 slides at a time. You can load those in. The system will go and do a pre-scan of those. You can select the regions that you want to image, tell it what channels you want, and then essentially just leave the system to go. So in our system, again, it's fairly low magnification, so 5, 10, 20, and 40x. These are all air objective lenses. I'll just show you one example from a group here. So again, this is the overview of the slide. We can use this to detect the specific bits of tissue. We can scan them at a very, very low magnification in fluorescence. And then you can use that to go in, say, if you want to image at a higher magnification, so 20x or even 40x magnifications on smaller regions. So the higher magnification, the larger the tissue, the longer the experiment. This is the type of system that you set up and leave overnight to run automatically. While we're talking about automated imaging, we have a high content imaging system. Again, this is a high throughput method. So samples are set up in 96 well plate, 384 well plate, whatever plate format you're interested in. So a plate rather than a dish. And again, the real power here is the automation. So we can tell the system to go to each of the wells, go to different number of positions within each of those wells, acquire in such and such channels, excuse me. And then the really powerful thing here is because we're acquiring so many images, you wouldn't want to pour through those images. So we can actually set up automated quantification. And at the end of the day, the readout from this system is more of an Excel spreadsheet than it is trawling through the thousands of images that you've created. So we can create plots here. So looking at drug responses and things within cells. So the pros on this is automated, unbiased, high throughput. So assay validation or screening. The downside is the optimization in terms of your sample prep and the image analysis, the massive amounts of data that you're producing. And also, again, because it's air-based lenses on this system, then obviously it's not very high resolution. The number of applications are endless. So there's a big long list on the left-hand side, which I won't go through. On the right-hand side, we can look at migration assays, so things like scratch assays, that type of thing. We can track cells over time, and we can look at morphological changes in response to drug selections or gene knockouts and things like that. So if you can think of it, we can probably pretty much come up with an experiment for that.